Hello, and welcome to Identifying and Mitigating Errors in Organic Extractables and Leachables Screening, presented by American Pharmaceutical Review and sponsored by Nelson Labs. My name is Mike Auerbach, Editor-in-Chief of American Pharmaceutical Review magazine, and I'll be the moderator during today's event. In this webinar, you'll learn about the three errors commonly encountered in extractables and leachables screening: the risk of omission, the risk of inexact identification, and the risk of inaccurate and imprecise quantitation in chromatographic screening processes. The presentation will explain these errors in detail and will address the issue on what can be done to minimize the occurrence of these errors or how to mitigate their impact on the final screening outcome. In addition, this event will hold a live question and answer session at the end of the presentation. To ask a question, click on the Ask a Question tab on the right side of your screen. Please take note, the right side of your screen also features an overview of this webinar and more information regarding our speaker. If you have a technical question during the event, click the Test Your Connection button at the bottom of your screen. From there, you can access additional webinar support. Finally, we encourage you to use the social media widgets beneath the webinar to share with your friends and colleagues. And now, allow me to introduce today's speaker. Piet Christians received his PhD from the Analytical Chemistry Department of the University of Leuven, Belgium in 1991. From 1992 to 1997, he was lab manager in two analytical contract laboratories. From 1997 to 2000, he worked as an independent consultant with Shell Chemical Company in Houston, Texas, where he conducted research on a new hydrogenation catalyst system for hydrogenated triboc copolymers. From 2001 onwards, Mr. Christians held the position of Scientific Director with Toxicon Europe, where he developed analytical methods and protocols for both extractables and leachable studies for the medical and pharmaceutical industries. In his current role at Nelson Labs Europe, Mr. Christians supports the strategy and global business development for extractables and leachable service offerings for pharmaceutical and medical applications. Pete, welcome to the webinar.
Well, thank you for the kind introduction, Mike. Today, I would like to talk about the errors one can make in the detection, identification, and quantification of organic extractables and leachables through the process of chromatographic screening. Now, chromatographic screening is an essential part of an extractable and leachable investigation of an extract generated from either a medical device or from a container closure system for pharmaceutical use. And what you're trying to do there is you try to make a total profile of your extract. It is a relatively new discipline. It's a new concept that has been introduced a couple of years ago. I think people who have been working in extractables and leachables for a longer time will know that concept. People who are relatively new to that industry may be a little bit in the dark of what it is about. For the content of the presentation, of course, part two will be the main focus of this presentation, where we will go deeper into the errors of chromatographic screening, which is the error of omission, the error of misidentification, and error of inaccurate quantification. However, if we want to talk about the errors of chromatographic screening, we need to define what chromatographic screening is, and that's what we will do in part one. We'll explain a little bit the process of chromatographic screening in that part. In part two, we'll discuss a little bit about the Nelson Labs Discovery and Screener database. When we're talking about the errors and how you can reduce the occurrence of these errors or mitigate the consequences or eliminate those errors, one of the main tools you can use is a database, and I will explain a little bit what we did at Nelson Labs to develop and populate that database. The first part is the chromatographic screening. What is it? Like I said, it's quite a new discipline. When you have generated an extract, whether it is an exhaustive extract or a simulated use extract or an exaggerated extract, what you want to do is to establish a complete extractables profile of that extract. You want to be sure that you have a complete understanding of everything what is in there. So step one in that process is you want to discover all those compounds, all those organic compounds. Of course, there is a multitude of compounds that potentially may come out of a material. You you will not be able to do that with one technique. You will need to take an approach of a combined orthogonal chromatographic approach. Once you have discovered those compounds, you may need to refine your search and say, we will not look at everything. We will only look at those compounds that are of toxicological relevance. And that's where you will use a toxicological threshold, such as the AET level. Everything that is above that AET level needs a further action. That's why the AET level is often referred to as an action level. The action is to identify the compounds and to quantify or semi-quantify those compounds. The identification makes the link to the relevant toxicological information of that compound, while the quantity of the compound in the extract will make the link to the dose to the patient that eventually will need to be assessed. Again, people who are familiar with analytical chemistry will mainly know the targeting approach, where you know upfront what you need to determine and what kind of compound you need to assess. In that respect, you're defining your target, you know the analytical method, or you select your analytical method and your procedures, and you try to be as quantitative as possible in terms of detection. In screening, however, you do three actions in a row. First, you try to discover all those compounds in the extract. The second action is the identification. Once you have detected them, once you have discovered them, you need to identify them. And in a third action, you need to quantify them. Another way of representing what chromatographic screening is you can compare it with net fishing. So when you're on your boat and you want to fish and you throw out your net and you move your boat and hope that you will catch as many fish as possible, you're depending upon the quality of your net. And as you can see here, the net is not really selective. You catch every type of fish. You can catch a bream, a herring, a catfish, a salmon, or a sprat. So you try to catch everything. And that is the same more or less what you do with an extract that is generated from a medical device or pharmaceutical container closure system. You throw out your net, which is your orthogonal chromatography method, and you try to establish an extraction profile that is as detailed and accurate as possible. Like I said, you use orthogonal analytical methods and you try to go for a broad detection. There are varying degrees of quantitation. We'll discuss that later on. However, there is that risk of omission that, as you can see, there are potentially small fish that will go through the net, will slip through the net, and you will not be able to detect them. On the contrary, if you're doing a targeted analysis, you can compare that with hunting. That means that up front, you know what you will be doing. You know, I will hunt a deer or a duck here in this case. And you know what the strategy will be. You choose your analytical methods. You optimize those methods. You develop them and you validate them. And that's how you can generate a very quantitative result. The downside of that is that you are only looking at those particular compounds that you have defined at the very beginning of your process. So you will not be able to see anything else than that. So how does a chromatographic screening process now look from a technical perspective? Here you can see a part of chromatogram of a GCMS, and everything that is above the AET line needs to be considered for further identification and quantification purposes. So what you do is, the first thing you do in GCMS is look at the mass spectra, which is more or less the fingerprint of every individual compound. And you look, can we say something about that? With GCMS, luckily, you can rely on mass spectral libraries like NIST and Wiley. There you can say, okay, based upon the quality of the hit, of the match factor, you can try to identify them. And in certain cases, you can say, okay, It is a confirmed identity. When we are looking at how USP is defining that, it is confirmed with an authentic standard. Or it can be a confident identification. That means that you have convincing proof of evidence that this compound is the true compound, is the right compound. Then you have a lower class of identification, which is the tentative identification. That means that you have an idea of what it is, but you're not really sure. Partial means you know some features of the structure of the molecule, but you don't know the complete structure itself. And an unknown is I cannot say anything. So based upon that classification, you can do your reporting, and you do a kind of semi-quantitative approach based upon, for instance, how the response of your analyte compares to the response of an internal standard. With this slide, I would like to explain why it is necessary to have orthogonal chromatographic approaches, so where you combine different techniques like Headspace GCMS, LCMS, and so on with different detectors. As everyone knows, there are a lot of medical devices, as well as that there are a lot of pharmaceutical container closure systems. Every single container closure system is made out of a certain material. And there's also dozens of types of materials like polyethylene, polypropylene, polyurethane, all kinds of materials that can be used in the manufacture of those devices. Not only that, you have different suppliers. Per material, for instance, for polyethylene, you may have 15 suppliers. For rubbers, you have 3 or 4 or 5 suppliers, depending upon the grade and the type of rubber. And every supplier has different grades. Depending upon the physicochemical properties of those materials, you can select a certain material for the design and the manufacture of your device. It goes even further. During the manufacture of your device, for instance, during the assembly, you may need to have processing materials or processing aids, or afterwards, your device may be sterilized, and that all has an impact on the extraction profile of those devices or container closure systems. That leads to a multitude of types of materials, of types of compounds that you can see. You can see polymer additives, you can see the degradation of those polymer additives, you may see oligomers, you may see slip agents, monomer residues, solvent residues. There's a multitude of types of materials, of types of compounds that you will see in those devices when you extract them with an organic solvent. And as you can see, if you look at those structures, they are very diverse. They range from small to very large molecules. They range from low boiling point, high boiling point. They range from high polarity to low polarity. That is exactly the reason why you cannot analyze all those compounds with one single technique. You need a combination of techniques, chromatographic techniques, to cover all those compounds as good as possible. It is estimated that the number of compounds that are there, the universe of extractables, is way higher than 10,000. We have about 5,000 to 6,000 compounds in our database. We know that we don't have everything, so it's way above 10,000. It will be somewhere between 10,000 and 100,000. And when you know that, you need to have some supporting tools to do the analysis, to discover them, to identify them, and to quantify them. And the solution that Nelson Labs has chosen is to develop a well-populated database to assist in that action. Now I would like to share a few words on the Nelson Discovery and Screener database, a little bit about the history, what it is, what is in there, and what kind of information is available per compound. Our development of the Nelson database started in 2006, and actually it started with the observation that when a material was analyzed for an extraction study, that we discovered that if you have a compound in an extract that it could as well be that if the flow of that project went one way in the lab, that it was reported as compound A. And when it went the other way, depending upon the expertise of the people who were working on the project, that if it went the other way, that it was reported as compound B. We thought, if you take yourself seriously, this is something that is unacceptable. So we said to ourselves, we need to create some kind of database to centralize all that information, to centralize the expertise of what the identity of certain compounds is. So we did some kind of inventory of all the studies that we had done so far at that time. And we started to purchase standards. Of course, if you would stop there, there is some kind of bias towards the compounds that you have detected already. So it was also our intent to make sure that compounds that are, for instance, in literature are also captured, or supplier information, for instance, what is on an MSDS sheet, or the known composition of certain materials. Of course, when you go to conferences, often case studies are presented or other things are presented, and we took all that information with us and made sure that everything that was presented was also in our database. We also looked at the regulations, for instance, packaging regulations, in particular food packaging, which often list a positive list of compounds that should not be in food packaging. We also went through that list. And of course, it's a continuous effort. It's not something that you do once. You do that every time. Every time you do a new study, you encounter new compounds, you buy those standards and you inject them and you add it to the database. The second step in the process is to try to purchase or obtain the authentic standard of the compound. The first step, of course, is look if it is commercially available. And since 2006, we have added 300 to 400 compounds a year from that perspective. But not every single compound is commercially available. You have seen the diversity of compounds, how broad it is. Well, sometimes we needed to synthesize certain compounds, or we needed to isolate a compound from the material that it was detected in. For instance, for the rubber oligomers, we did the isolation from the elastomer itself. Once you have obtained all those standards, you need to analyze them. You need to analyze them with the orthogonal chromatographic screening methods. Of course, the first thing you do is look at what was the method where we detected this compound, and you choose that method to do the analysis on. However, for instance, for NVOC, it is very crucial to build your knowledge base, and it's often the case that things that were initially detected in the semi-volatile compounds analysis, for instance, we also analyze with NVOC, and we see a compound that was reported as an unknown before, all of a sudden becomes an unknown. Step 4 is the preparation and analysis of the analytical standards. So first step of that is the standard preparation. So you purchase your authentic standard, you make a primary standard out of that, and then a working standard to inject it in a second step into the instrument. And then you're capturing all the information that is generated, such as retention time, mass spectrum, and the relative response factors. Relative response factors relative to the internal standard. And that is how it's looking like. This is an excerpt of a list of compounds that is in our database for GCMS between retention time 19 and 19.2. You can see the detail here of what is in the database. I'm not saying that it's an Excel spreadsheet. It's actually a database itself. However, you need to export some of the information if you want to show it. But what you can see here is that we record retention time, the name of the compound, the CAS number, the relative response factor, which is very important if you want to do more quantitative assessments, and then mass spectral information, such as the target mass and the qualifier ions and the ratio of the qualifier ions that you should detect them in. And that is the status here of where we are right now with the database. For Headspace GCMS, we have over 1,000 compounds in that database, of which 95% are confirmed according to the definition in the USP. And that is the status of the Nelson database as of June 1st, 2020. So there are about 1,000 compounds in the Headspace GCMS database, of which 95% are confirmed according to the definition of the USP. For GCMS, we have over 3,600 compounds in the database, of which 78% are confirmed identity. And for LCMS, combined APCI positive or ESA positive and negative, we have over 2,000 compounds in that database, of which 1,500 compounds are based upon authentic standards. And now we'll come to the actual subject of this presentation, the errors one can make in chromatographic screening for extractables. And the first slide gives a little bit an overview of what kind of errors you can make. Actually, there are 4 errors. However, we will only focus on 3 in this presentation because the 4th one would bring us too far, and that would take a little bit too long. We will focus in this presentation on the error of omission. That means that you may not have detected all compounds that were in your extract. The error of misidentification. That is, once you have detected it, you have given it a wrong identity. That means that it has a wrong structure, a wrong name, a wrong gas number, and it has also consequently a wrong toxicological evaluation. A third error is the error of inaccurate quantification. That means that you have detected it, you have identified it, but you gave it the wrong concentration. And the fourth error is the error of implementation. That means that you have developed the methodology, but you were not successful in the implementation on a day-to-day basis. So you need to qualify your methods, you need to determine linearity, you need to determine LOD, LOQ, and during every run you need to do some kind of quality control, such as a system suitability test. Like I said, number 4 in these errors will not be addressed today. These errors are also described in 3 scientific articles that have been published in January in the PDA journal. The references to those articles will be given at the end of this presentation. We'll start with the error of omission. So what is it? It is the inability to detect all those extractables at or above a justified reporting or safety threshold, for instance, the analytical evaluation threshold. The consequence of that is that those undetected extractables will not be able to undergo an assessment. The severity is a fatal error. When you have missed a compound, the extraction profile is irreversibly compromised, and the potential toxic compounds will be missed, so wrong conclusions on safety will be drawn then. There are typically 2 errors one can make in the error of omission. It is falling through the cracks, and it is failing to see the tree in the forest. When we first look at falling through the cracks, that first error, you can subdivide it into different sub-aspects of the analytical methodology. First of all, some compounds may never have made it to the column, for instance. I think most people who have been working with GCMS or GC in general know that some compounds may stick to the injector, may not come off, or sometimes something happens in the injector, for instance, degradation, or sometimes there's some kind of ester formation, an undesirable conversion into an ester. These are all things that you do not want to take place. Another thing that can go wrong is that something unfortunate happens while the compound is in the instrument. For instance, if the solubility of your target compound is not compatible with the elution solution, for instance, it may precipitate. Or compounds may be eluting into the solvent peak, or in general, you could say that coelution may be a problem in certain cases. It is also possible that your compound may never come off your column, and that is when you have a stationary phase of the column that is not suited for the analysis of the particular compound, that the polarity of the stationary phase does not allow a certain compound to come off. It sticks onto the column. And of course, number 4 in those errors is that something unfortunate may happen with the detector. And that is particularly true for LCMS. It is very likely even that some of the compounds will not be ionizable in one of the modes that you select for your LCMS. That's why you need to consider more detectors for LCMS than only one, like APCR positive, negative, ESI positive, and ESI negative. That's our approach. Further details on the error of omission you can find in part 1 of the series of scientific articles that were published in January in the PDA journal. The second type of omission error is failing to see the tree in the forest. And that can happen when you have a loaded or overloaded chromatogram, as you can see one here. It can happen at the level of an extraction study, where you made an exhausted extract with a high number of compounds at high concentrations, for instance, or it can happen at the level of a leachable study, when you're determining the concentration of a leachable in a drug product, for instance. What you're actually doing is you try to kind of visually compare a blank with the drug product here, in this case, that had contact with the container closure system, and you try to visualize or visually compare those chromatograms and try to assign every single peak. But as you see, it's not always very easy. Certainly not if you're doing that on a visual basis. Of course, there are now software tools to do that, to assist in that. But when your process is based upon visual inspection, there is a higher probability that you will miss certain compounds. How can a database reduce the errors of omission? First, by injecting more than 4,500 authentic standards. We know the responses of every single compound in the certain mode that it has been analyzed in. So we know what we can detect with our orthogonal chromatographic methodology. However, we also know what we cannot detect. So there is also knowledge on the compounds that potentially may be missed in a screening study. And that is crucial information, because that allows you to optimize your methodologies. If your standard combination of techniques is not sufficient to detect certain compounds based upon the knowledge of the material and the composition of the material, you may need to expand your analytical strategy. A database also enables to perform targeted screenings, because all the information that is in the database on the compounds, like retention time, mass spectrum, allows you to do to zoom in into the chromatograms and look for specific information at these retention times and look for certain mass spectral features for that. So that allows you to avoid missing certain compounds because you did not see the trees in the forest. A second error is the error of inexact identification. So what is it? It is the inability to identify all compounds at or above a justified reporting threshold, such as the analytical evaluation threshold. The consequence is that there is an inability to link the compound's identity to the associated toxicological consequences of that identity, and the severity of an inexact identification is that it's a fatal error. An inexact identification precludes a proper safety assessment, and I will explain that later on how that looks like. Now, in this first slide on the error of identification, I would like to explain what is the most commonly used methodology in identification for GCMS, and that is mass spectral matching. So you have detected a compound in GCMS, and you have detected that mass spectrum. So the first thing that is often done is you look at the mass spectral libraries such as NIST and Wiley, and you come up with a mass spectrum matching list. So that's how it's looking like. Either you compare it with the structures in this case, and it is typically ranked from a high match to lower match. You could also rank it more in a table form, where you have the match and the reverse match and the probability score and so on. What is often used as a practice is to report the highest match factor as the true identity. And what I think is not really the best option, although it can be that the highest ranked compound is truly the right identity. It really depends upon the quality of fit. And if a compound is not in the database, even the highest ranked will never give you the right identity. I will show you some examples. I will show you 4 examples where the strategy of mass spectral matching goes wrong. And that is the first one here. So this is a compound that is detected in a GCMS analysis, and mass spectral matching gives this result with this structure, while we know in our database that it is this structure. We know that because it has been run as an authentic standard. We know its retention time, and we also know its mass spectrum. And you can see that the fit of the mass spectrum is way better than what was given as an identity in the NIST or Wiley library. Most probably this compound is not present in the NIST or Wiley. In this case, it's a rubber curing accelerator. Here is another one. So this is a mass spectrum that we detected also in the study. This gives you the highest rank in terms of mass spectrum. The mass spectral matching is the highest one here reported. And you can see that it gives a library match of 70%. Well, 70% may give the wrong perception that we're talking about 70% of correctness of identification. Well, it is not that. It is the 70% is the how good the resemblance is between the mass spectrum of the reference mass spectrum compared to the target mass spectrum. Also, what you can see is that with the first identification, the one with the highest match score, you can see that the resemblance visually of the mass spectra is really not great. So you know that something is going wrong here. I mean, if you simply would report this compound, you know upfront that it may not be the right compound. The compound that we found being present in the Nelson Labs database here is this compound. It's a polyolefin catalyst that is used in the polymerization of a polyolefin. It was mentioned before that identification errors are fatal, and with this slide I would like to explain why that is. So you can see on the left-hand side a chromatogram that calls for some action. The first thing that you can say about that chromatogram with the 3 peaks is at least those 3 peaks were discovered. So they were discovered, and you, based upon the level of the AET, where it is, where the line is drawn, you can say 2 of those 3 peaks need to be further identified. When things go right, and you have chosen the right strategy to identify and you came to the right conclusion, you can say, okay, these are the structures that are associated with those peaks. And that leads to this kind of information, toxicological information like TDE, TIs, reprotox, systemic toxicity, and so on. However, sometimes things are not going well, and you may miss the identity of certain compounds. In this case here, you can say, okay, you can clearly see that the structures are not the same as when it is the right ID. So you're basing your assessment on the wrong structures, and it leads to a wrong set of toxicological information. And that is fatal, because there's no way back. There's no one who will say at this point, your identification was not correct, so we cannot use that information. So that means that it has consequences for the safety of your device or of the quality of your assessment, your toxicological assessment. Now, what is worse, a misidentified compound or no identification for the compound? A misidentification, and that is typically happening for GCMS, is considered as being worse because in a lot of cases, there's no way back. There's no subsequent correction for a misidentification. Reporting a compound as an unknown is something that you will typically see for LCMS. Of course, you cannot do an in-depth assessment also for a compound where you don't know the identity of, but at least you know that some additional action need to be done. You need to do your homework and try to come up with a better identification using higher-end analytical techniques. Now, what is happening if we, with the database, if we do a study and we see a compound that is not in the database yet, or a compound that we have never discovered and we do not have a clue what it is, we use, first of all, the high-end analytical techniques such as GC-QTOF, accurate mass-based GCMS, or the exactive Orbitrap of LCMS. Based upon that information, and typically it's a combined combination, you try to identify the compound, you purchase the standard or you synthesize the standard, you analyze it again, and you record retention time and mass spectrum, and you verify if it's the same retention time and mass spectrum as you saw with the unknown, and you put it in the database. So that's the way you try to eliminate the unknowns and try to build your database with known compounds. Now, what about LCMS identifications? We talked about GCMS identification strategies and mass spectral match factors, but what about LCMS? Well, there is no NIST or Wiley for LCMS, and in a lot of cases, this is the result that you will have from an LCMS analysis if you do not have a database. So you report a long list of compounds. All those compounds will most probably unknown. You may report also some kind of other information like retention time, maybe the mass that you have seen it at, and also the concentration or indication of concentration. Now, what can you do with a database is, this is the outcome if it has been run through the Nelson database. You can see that there are a number of compounds that have been identified. If there is a CAS number here, that means that we have the authentic standard. If it has a tox number, it means that we know what it is. It is in our internal database with that number. However, we do not have the analytic standard. So it's present as a tentative or most probable compound. Having that information coming from the database also allows you to go further. I mean, you can see here that 5 or 6 of the compounds were still unknowns. What you can do with accurate mass LCMS, like exactive Orbitrap or Q-exactive Orbitrap or any other QTOF LCMS, is you can look at the elemental composition of those compounds. And here, with that information, in combination with the information that is already available in the database, the compounds in the database provide you certain information already. You can say, okay, the ones that we additionally identified most probably are diamides, because we saw other diamides already being present and that were positively identified. And the elemental formula fits that structure. So how can the Nelson database reduce errors of misidentification? All the compounds that are in the database and that were confirmed via an authentic standards will receive the confirmed identity, and that is in line with the definition of the USP 1664, where mass spectral information and retention time match is required. However, we also have other information in the database that is not based upon authentic standards, have a lower degree of documentation, but still we are relatively confident for certain compounds that we're looking at the right structure, or it is a tentative structure. Now, the error of inaccurate quantification, that is the last error we will discuss, when the quantification of extractables is insufficiently accurate. The consequence of that is that the assessment of the exposure of an extractable to a patient, for instance, through the evaluation of PDEs, will be compromised and may lead to flawed conclusions. Now, the severity of the error of inaccurate quantification is that it's a critical error. The safety assessment can be performed, but the inaccuracy of the concentration or dose needs to be accounted for. And that is something you can do via, for instance, in the introduction of uncertainty factors. It is practically impossible to have calibration curves for all compounds, all extractable compounds in an extraction profile. Like I've said, it is very likely that the number of extractables is anywhere between 10,000 and 100,000. It is practically impossible to have a calibration curve for all those compounds. So you need to have other approaches that allow you to estimate the concentration to a level that it allows you to do a proper safety assessment. Let's first look at the simple quantification, and that is the practice that most labs are using to quantify extractables in an extraction profile. You can see a chromatogram here with two equal peaks, and when you're trying to determine the quantity of those compounds, you're in a comfortable position already because you have avoided two fatal errors already. You have discovered the compounds and you have identified it. The only question now is how much of that compound is present in an extraction study. So with simple quantification, you assume that the response factor of the internal standard, which is referred to as IS here, is the same as the response factor of the extractable. So what it means is that you add the internal standard to your extract at a known concentration, for instance, 10 milligram per liter, and it gives you a response in that chromatogram of, let's say, 100, an area of 100. You can see here that the extractable has the same area as your internal standard. So here you're assuming they have the same signal, so most probably they will have the same concentration. So the extractable here will also most probably have 10 milligrams per liter as a concentration. Or that's what you assume. That is what you assume in a simple quantification. Of course, you can further build in some uncertainty factors to make sure that that simple quantification is that the inaccuracy of the quantification is accounted for through the uncertainty factors. Or you could use also surrogate standards, for instance, where you say, okay, this compound I have identified, it looks like an aldehyde. Let's take an aldehyde as a comparison and use the response factor of a surrogate standard that is an analyte to correct for the concentration. Now, if you use surrogate standards to correct for responses, you have to be prudent. You have to select the right rationale why you want to correct a certain compound with a certain surrogate. For instance, as a rationale, you could say the compound was identified as an aldehyde, so let's take an aldehyde as a surrogate to correct the response. That may be oversimplified, and I will try to explain that with the table. What we did with our database is try to split it up, the entries that we have in our database between the different functional groups. So we have esters, alcohols, and ketones, and so on. And if you look at the statistics, at the mean, the standard deviation, the median, the minimal response, and the maximal response, you will see that aldehydes behave quite similarly to any other group of compounds. So it is not a good predictor to take one aldehyde and say, okay, we will correct all the other aldehydes to account for the uncertainty of the quantification for that aldehyde. Let's now look how we can upgrade the quantification from a simple quantification to a quantification using relative response factors. Typical information that is in a database if you have analyzed the authentic standard. So in step 1, you determine the relative response factor for the extractable compound. You analyze the extractable standards at a level of 10 ppm, which is the same as the concentration of the internal standard. But you see, the response is different. You see, when you look at the chromatogram, you see that the extractable only has 1/3 of the area compared to the area of the internal standard. The concentration of the internal standard is 10, but also the concentration of the extractable is 10. So you can say, okay, however, the areas are different. The areas of the internal standard is again 100, while the area of the extractable is 30. And that's where you determine the relative response factor, the response factor relative to the internal standard. Here, in this case, is 0.3. And that is information you put in your pocket and you use it when you encounter that extractable again in another extract. And that's how it goes. So again, you see a chromatogram where you see the same extract as the one that you have determined the response factor of. You have added the internal standard at 10 milligrams per liter again, and it gives an area of 100. The area of the extractable you want to determine the concentration of here gives 100, but you know the relative response factor here for that compound is 0.3. So the concentration, instead of with a simple quantification where you obtain the concentration of 10, here the concentration appears to be 33. Which is much closer to the actual concentration or the true concentration than the 10. In our database, we have this kind of information for over 4,500 extractables, so we can do that correction for all those compounds that are in the database. A little bit about the information that also can be found in the scientific articles that have been published in the PDA journal. That table gives a little bit an overview of the compounds that are in there, the number of compounds we have, relative response factors of the mean, the median, the standard deviation, minimum and maximum RRF. This information can eventually be used to calculate uncertainty factors, for instance. What can be seen also from the previous table is that the range or the standard deviation for GCMS is much more narrow than for LCMS. The probable value range when the true value of a concentration is 1 in GCMS, typically it will range between 0.5 and 2. However, there are certainly compounds that will fall out of that range also. For LCMS, when the true value of a concentration of a compound that is detected in LCMS is 1, you will see that relative response factor varying between 0.2 and 5. When you use the statistics of the database, you will come with uncertainty factors for LCMS that are way above 5, closer to 10, and sometimes higher. So that is not usable. That you cannot use those uncertainty factors to correct, because that will not be practical anymore. You would need to analyze or evaluate your chromatograms way too low. What we chose to do at Nelson Labs is to do an individual correction of each and every entry that is in the database for LCMS. That way, we do not need to work with uncertainty factors for the compounds that are in the database. For GCMS, because the range is much more narrow, we still can use uncertainty factor and calculate those uncertainty factors and use them to address or to calculate the AET level and to correct the reported concentrations. Of course, one could ask himself, how well does a one-time determined relative response factor compare to a concentration that is obtained via a full calibration curve? You know, you only do that once, you only inject all those standards once, you obtain that response factor, and you would use it in every further evaluation. And how does that compare to a concentration that is obtained via calibration curve? And that's an exercise we did using the compounds that are in our system suitability test. This is the list of compounds that is in our system suitability test. And we compared the RRF factor with the response function that was obtained through calibration curve. And you can see when you look at the percentage of deviation between the database RRF and the response function RRF, you can see that although there's some variance, for most of the compounds, it's a very good estimate into the true concentration. There's one exception, and that's 2-mercaptobenzothiazole. However, this is an LCMS compound. We have that information also in our GCMS, but typically we analyze and report it as an LCMS compound. So in this case, in this evaluation, it was less of a relevant compound. Now, how does the Nelson database reduces error in inaccurate quantification? Because of the fact that we have all that relative response factor information of those authentic standards, we can screen in a quantitative way. And the relative response factors can be used in two different ways. We can, at a minimum, calculate the uncertainty factors based upon the statistics of the database. And in the second application, we can correct, for instance, in LCMS, every individual compound with its associated response factor to have a better estimation of the concentration that way. We think that also RRF correction is superior to a surrogate correction, which is a practice that is referred to in the ISO 10993 Part 18. We think you have a better estimation of the concentration using individual RRFs, because at that, it's much more neutral, and you don't need to rely on expert judgments on what kind of surrogate to pick to correct for your responses. Concluding remarks. First of all, although chromatographic screening is the only testing strategy that fills the need of a good E&L assessment. It is prone to errors, and we have discussed in this webinar the three most occurring errors, and that is the error of omission, where you may miss compounds, error of inexact identification, and errors of inaccurate and imprecise quantitation. And I hope I was able to convince you that the Nelson Labs database, which is part of our strategy, is able to mitigate, reduce, or eliminate some risk of occurrence of those errors through the use of that database. For more educational materials, such as webinars, white papers, articles, and other information, I would like to refer to the Nelson Labs website, where you will find a lot of interesting stuff on sterility testing, biocompatibility testing, in vitro tox, chemical characterization of pharmaceutical container closure system testing. But I would like also to refer to the Sterigenics website. You know, Sterigenics is a sister company of Nelson Labs and specialized in sterilization. So you'll find a lot of valuable information on sterilization and sterilization validations on their website also. And as a last slide, these are the references to the scientific articles that you can find in the PDA journal for people who would be interested. And with this, I would like to conclude my presentation and open the floor for questions.
Thanks, Pete, for your
much.
very informative presentation. I'd like to remind everyone that you can submit any questions for Pete in the Q&A box on your screen, and we'll get to as many as we can. So let's now take a look at our questions. And our first question is, in the error of misidentification, you said that for GC-MS identifications, the use of NIST and Wiley mass spectral libraries leads to a lot of errors in identification. However, it is a practice that we and many others use to identify compounds. Can you be a bit more specific in what exactly is the problem with these identifications?
Well, Mike, there's nothing wrong with those libraries, nor in the use of those libraries to try to identify those compounds. Where sometimes things are going wrong is how these libraries are used. And typically you have the ranking of the compounds based upon a certain mass spectral match factor. And if you only consider the highest ranked compound, then there may be a problem. And the problem is there because not all mass spectra of all compounds in the world are in there. Sometimes for those compounds that are in the mass spectral database, the highest ranked will most probably also be the compound. But for those compounds that are not in the database or in the NIST and Wiley libraries, if you then rely on the highest ranked compound, you will for sure misidentify that compound. So what we always advise to do, and what common practice is in our lab, is to always, even if you have a high probabilities, high mass spectral match factors, even then rely or use an expert advice of a mass spectrometrist to be sure that you come to the right conclusion in terms of identity, because it is so crucial to have the right identity. It's only then that you can link the identity to the relevant toxicological information.
Okay, terrific, thanks. Let's move on to our next question. Is it possible not to perform the leachable studies when there isn't any compound above the AET in the extractable analysis?
Well, here we're talking about extractables and leachables for pharma, for drug products. There are different angles to that question. Historically, we're talking about 10, 15 years ago, actually, it was not really frequent that leachable studies were performed. Most people relied on the results of an extraction study, and extraction studies were considered to be done under harsh conditions and should be representative of all compounds potentially that should be leachables. So in that case, not a lot of compounds were, or in not a lot of cases, the next step was taken to leachables. However, we have learned that not all leachables are a subset of extractables, certainly not in the pharma space. There are some compounds that we see as leachables that you can not always link to the extractables that you have seen at the level of an extraction study. That's why also the authorities and regulators want to see actually the results of a leachable study. Now, in the case that you have performed extraction study and there is really no compound above the AET level or any threshold of toxicological concern, you definitely can limit your efforts at the level of a leachable study. But our advice is, if you assume that you will not see anything based upon the results of an extraction study, prove it with a short study that your assumption was right. Like I said, I'm not talking about extractables and leachables studies for pharmaceutical drug products. I was not talking about chemical characterization.
Okay, thanks, Pete. Our next question is, in using RRF, is the expectation that it is reconfirmed at time of use to account for differences in the analytical instruments versus the one where RRF was initially determined, and instrument MS detector tuning and responses on day of use?
That is a very good question. It is, of course, impossible if, I think in total we have something like 4,500 compounds where we have the RRF data from, across different chromatographic platforms. So it is simply impossible to do that check every time you run a batch of analysis. What we do at Nelson Labs is we have a system suitability test that is something like 10 to 12 compounds. And we run that on every platform. So every platform has its own selection of system suitability compounds. And we run those compounds at the beginning of an analytical run, in the middle and at the end. And we verify some chromatographic and mass spectral information. And one of those things is to check if the response factors or the relative response factors are within certain ranges as we have seen them during the initial injection of the compound of the authentic standard. So it is verified if the response is going well for those compounds in the system suitability test.
Our next question is, what is the formula that Nelson used to calculate AET for medical devices, and how does Nelson calculate an uncertainty factor for use in AET calculation?
Well, the formula itself, it's a bit difficult to explain it, without being able to visualize it. But it contains the factor of the threshold, the dose-based threshold. It contains the information on the volume of the extract. If you have prepared your extract, you do it in a certain volume of solvent. That plays a role. Also, the number of devices that were used to make your extract will be taken into consideration, and the number of devices per day that will be used to treat the patient. This is basically, these are the variables that are in the formula. It's difficult, like I said, it's difficult to explain that formula. Of course, the AET is then further corrected with an uncertainty factor. And the uncertainty factor that was in the ISO standard originally was used by us to indeed to calculate the uncertainty factor for our methodologies. However, recently there has been a communication that the formula that was in the latest version of the ISO 10993 part 18, that there was some kind of inaccuracy in it, and it's now 1/1 minus the RSD that is used, and that is also the formula that we're using in our calculations.
All right, let's move on to our next question. Have you developed a universal screening method depending on the material you need to analyze?
Well, like I said in my presentation, the database itself is based upon all the observations that we have done across all kinds of devices, across all kinds of materials, with all kinds of inputs of regulations, of literature, of research that we did ourselves. So the database itself is not really optimized for one particular material. However, if we know of certain materials that they will be part of the device or subject to the extraction study, and we know that some compounds in that material may not give good responses in our methodology, then we may need to augment our orthogonal chromatography with certain techniques. I will give you an example of one compound that is formaldehyde. If you have a POM material, for instance, polyoxymethylene, it is known that formaldehyde will be present. We know that our methodology for formaldehyde is relatively low in response, and we need to augment those analytics with more targeted analysis for formaldehyde.
Thanks for your answer. Let's move on to our next question. Is there a general advice you can give in how you can select surrogate standards to correct for the quantitative estimate of the concentration of the extractable?
Well, per definition, so surrogate standards are those standards that are used to calculate the concentration of your extracts. For us, the surrogate standard in our methodology is the internal standard, because we use the internal standard and the response of the internal standard to correct via the RRF correction, to correct for the response factor variation, and come with a more accurate reading of the concentration of the extractables. In some cases, there is also a practice where some compounds, a mix of compounds is used, like 5 to 10 compounds with different functionalities, different sizes, different boiling points, or different polarities, or whatever. And that based upon the structure that you have identified for your extractable, that you say, okay, this compound is very close to the structure, or close enough to the structure of that surrogate, so we'll use the response factor of that surrogate compound and correct with that response factor. With that practice is depending upon also, let's say, the quality of the judgment on how you can do your assessments. I mean, for some compounds, it will be obvious which kind of surrogate to take, but you have some extractables, for instance, so they have two types of functionality. So it is sometimes difficult to know which functionality to take, and that would be the most representative. Also, we have seen in our analysis of the database that, for instance, the size of a molecule will play a role. So, for instance, if you have an alcohol, the smaller alcohols will give relatively low response factors. The higher molecular weight alcohols will give higher response factors. So it's not because it's an alcohol that you could say, okay, I have an alcohol here, I detected an alcohol, so I will take an alcohol in general, because that will not work. You need to be more specific on the features that you select in terms of reference.
Okay. Moving on to our next question. If you want to build a database or identify compounds in LC-MS, is it always necessary to have access to high-resolution, accurate mass instruments?
There are two components to that question. First of all, is it necessary in general when you perform extractable and leachable studies? Is it necessary for LC-MS to be accurate mass, regardless if you have a database or not? I think it is. I think the power of identification of high resolution accurate mass is substantially higher, is way higher than unit mass. That doesn't necessarily mean that a high resolution accurate mass instrument automatically will give you the identity. However, it will give you that kind of information that allows you to explore further what a compound could be. That could be through the evaluation of the elemental formula, and combined with some of the fragments, for instance, you could come to some suggestions of structures, and maybe in combination with what you have detected with GCMS or with derivatization or with other techniques, you can come with some kind of suggestion of what a compound could be in LC-MS accurate mass. Now, the second part of the question, is it necessary if you build a database that it would be in accurate mass compared to unit mass? Well, our experience is we started to build our database first in unit mass. That was in 2009, and we came to the conclusion that once we had more than 200 compounds in it, that unit mass was not specific enough anymore, and that we started to have some inaccuracies in the identification. So for us, it was clear that you could only build that kind of database for LC-MS in a high resolution accurate mass environment.
Okay, let's take a look at our next question. Do I understand that your RRFs are based on the internal standards selected? Is it then a common practice to use the same compounds as internal standards for all studies?
Yes, so what we did is we always use the same internal standard. We have our internal standard for Headspace GCMS, one for GCMS, one for LC-MS positive, APCI, and one for APCI or ESI positive and negative. We always use the same internal standard at the same concentration because we have done in our evaluation, we have evaluated the response factor of every analyte of the extraction compared to that one particular internal standard. So we know exactly what the relative response factor is for that one internal standard. So we don't need to inject 5 because that would not give us additional information. That one internal standard gives us exactly what we need in terms of reference towards the relative response factor. Okay.
So the next question is, if you perform such studies, do you encounter additional issues with screening for leachables due to product matrices?
Well, that can happen. Absolutely. It can happen that depending upon the composition of your drug product and the complexity of its composition, that you will see that some of those leachables will be masked, their presence will be masked by other ingredients like excipients or antioxidants sometimes, or the API or degradants. So that happens. The advantage is, and that is seeing the forest through the trees, the trees through the forest, I'm sorry, is that with a database, you know exactly what you're looking for. For all those compounds, for instance, in GCMS, we know at which retention time we need to look for which kind of masses. And that, of course, narrows down a lot of your analytical work. So for all the compounds that are in the database, we can jump below those large peaks and look below those peaks what is in there for those targets that are in the database. It is a different story, of course, if the compound is not in the database, the sensitivity of the method will be substantially higher.
Terrific, thanks. So unfortunately, that's all the time we have for questions. Any unanswered questions will be answered via email. We'd like to thank Pete Christians for sharing his knowledge with us and also offer a special thank you to Nelson Labs for sponsoring today's event. Please keep a lookout for an email containing a link to view this webinar on demand and also to share with your colleagues. Thanks again for joining us and enjoy the rest of your day.